group at the University of Utah in Salt Lake City, and this is just my email address. If you ever feel like contacting me, feel free to. I recently started this position right before we had this pandemic season start, and before that, I was a graduate student at the University of Texas at Austin, and that's why I have this logo right here. In addition to that, I have Skoltech, which is the Skolkovo Institute of Science and Technology, where my PhD advisor actually moved. This is all the way in Moscow, Russia, and it's a pretty awesome graduate level research university that he helped build. So today I will talk to you about the real-time electrochemical detection of pathogenic bacteria and give you a broad overview and summary of the most important findings of my PhD work from UT Austin. So before I start, I'd like to give you a little bit of background about myself. I am originally from Skopje, Macedonia, which now is been renamed to North Macedonia. So if you hear a little bit of an accent, that's where it's coming from. In 2011, I moved to upstate New York to attend Bard College, where I graduated with my bachelor's degree in chemistry. Then I moved down to UT Austin in 2015 and graduated with my PhD in December of 2019. And now I'm at the University of Utah in Salt Lake City, as I said earlier, in the Mintier group. So I just wanted to put this a little avatar my friends made of me for my 25th birthday. This is supposed to look like me, though it doesn't really look like me. And in the background, you can see a circuit of bacteria, basically. So that's kind of, it's supposed to describe, it was meant to capture my entire PhD work as my friends saw it. So I thought I'd share that a little bit about myself so you know what my research journey really has looked like. So we live obviously in unprecedented times and where we have this global pandemic. And I just want to remind you all that pathogens are everywhere. They're in our hands, in our mouths, on our skin. I'm sure that many of you have been constantly seeing this representation of this corona type viruses. And today I will talk more about the pathogenic bacteria species, which are this rod-shaped creatures. So first, I'd like to start with telling you about the motivations and challenges to detect these pathogens in very, very complex and dynamically changing environments, which really motivated my PhD work and kind of provided the fundamental questions that I wanted to explore. So bacteria, typically pathogenic bacteria, you can think of them as highly social microorganisms. So they're very much like humans in a sense, in the regard that they tend to organize themselves into this small groups. So what you can notice here, I'm showing a very simplified schematic where there's 3 distinct types of pathogenic groups that are forming, and these are representing immune cells. So each of these communities actually are able to develop their own sort of, to speak, language of biogenic signals and toxins, which they use to communicate with each other in these communities, as well as with the neighboring communities that are around them. These biogenic signals and toxins can vary largely in sizes, going from very large molecules such as cytokines, hormones, neurotransmitters, and sugars, all the way down to very small and highly diffusive molecules such as hydrogen peroxide and nitric oxide. In addition to all of this, there's a lot of environmental factors that can impact the lives and the ways in which these cellular communities develop and live. This includes signals, nutrients, pH levels, oxygen levels, temperature, pressure, and many others. So we're looking at a very complex system here. The most typical way to study this cellular communities, especially in the world of microbiology and even in chemistry, is to use fluorescence-based microscopy techniques. So here I'm just showing you a example image of a confocal uh, taken at a confocal microscope that shows you 8 types of bacterial species that are present into your oral microbiota or your mouth. So you can think of these as kissing bacteria. Um, Basically, your mouth contains over 1,000 species, and these fluorescence-based microscopy technique provides a very good visual representation and this pictorial image of what kind of species you have in these environments, how they're spatially organized, and how they tend to group themselves into these distinct groups and which neighboring communities are around them. This technique is rather very static. It's not necessarily real time, and it does not provide ways to quantitatively study these environments. In particular, there's a lot of time delays that are associated with these fluorescent proteins. Therefore, it's not a technique that allows us to study these complex environments in real time. So how can we develop a platform that allows us to study this complex and dynamic environments in real time in a quantitative manner? To do that, we're going to use electrochemistry and a very specific system that was part of my PhD work. So before I tell you more about the electrochemistry, I'd first like to introduce this species that I worked with during my PhD. It's called Pseudomonas reginosa. And Pseudomonas aeruginosa, it's actually an opportunistic human pathogen, which can cause you great harm if you get it. So this species is majorly responsible for about 80% of hospital-acquired infections in the world. It's also the most commonly isolated species from people who are suffering from chronic wounds and people who are suffering from cystic fibrosis. So this is actually the major pathogenic species that contributes to the cystic fibrosis development in the human lungs. This is just a microscopy image that was taken by my biology collaborators, the Whiteley Lab, which used to be at UT, but now are at Georgia Tech, where you can see better what this bacteria looks like, very typical rod shape, and it has this elongation protein, which it uses to basically move around. So Pseudomonas aeruginosa, it's actually particularly interesting to us electrochemists, mainly because it produces this bacterial warfare toxin, which is also considered a virulence factor called Piocyanin. Piocyanin actually helps Pseudomonas aeruginosa spread infections in the host organism really easily, and it's produced by the bacteria itself during very early stages of infection. In addition to that, Piocyanin is exclusively produced by Pseudomonas aeruginosa, so no other species produces this factor, and as such, it's an effective diagnostic biomarker that allows us to identify infections that are specifically caused by this pathogenic species. So we're actually very lucky that Piocyanin and Pseudomonas aeruginosa in general, it's considered an electrochemically active bacteria because it produces this electrochemically active species Piocyanin. This shows you the redox reaction of Piocyanin, where you can see the reduced and the oxidized states and the transfer of electrons. This is actually a rather complex electrochemical mechanism, as it doesn't involve a pure electron transfer. We also have bimolecular steps that accompany this electron transfer process, and that involves the transfer of these protons. Because this molecule is actually electrochemically active, we can use electrochemical methods which provide fast and direct detection. So there's been a lot of interest in the electrochemistry community in developing sensors that are specifically aimed at detecting Piocyanin as a marker for understanding virulence mechanisms of Pseudomonas aeruginosa. However, something that I've really focused on and sort of accidentally discovered during my PhD work is that this Piocyanin is actually only the end product of a larger biosynthetic pathway in Pseudomonas reginosa, which is known as the phenazine biosynthetic pathway. So this shows you a very simplified schematic. It doesn't show all the biochemical conversions and steps, but it starts with chrismic acid and it goes to this PCA species, and then goes to 5-MCA. And finally, we're at Piocyanin as the final product. I should mention that in addition to this, there's other decomposition products that can form from Piocyanin, depending on what pH conditions you're working with. So just to clarify what phenazines are, if you can see already from this ChemJAWS provided here, these are this heterocyclic nitrogen-containing compounds which have subtle differences in the functional groups around them, and they're all actually electrochemically active. So we can use electrochemistry to detect not only the final product, but really detect this other species that are part of this biosynthetic pathway, some of which we're going to see are actually rather highly reactive. So as such, we get a better image in real time of what's happening as Pseudomonas aeruginosa grows and develops infections. So before I get into all this real-time data that I will talk a lot about, I first wanted to tell you about the biological sensing platform that all these experiments were done on. It uses transparent carbon ultramicroelectrode arrays, or TCUAs. These electrodes were developed in the Stevenson group, and they were actually a great idea of my PhD advisor. So it's basically an array-based electrochemical platform where we have a quartz slide with a pyrolyzed photoresist film on top, on which we then apply polystyrene spheres. And upon that, we then applied 10 nanometers of alumina oxide layer, after which we melt the polystyrene spheres, leaving us with this array of ultra micro electrodes where we have these exposed carbon areas where the polystyrene spheres touched our pyrolyzed photoresist film, and the rest of the electrode area is governed by this aluminum oxide layer. So because of this geometry and basically, if we look down into the capacitance and the noise and the signal that contributes to this sort of electrode geometry, we get various advantages, including enhanced analytical sensitivity, improved signal to noise ratios, low limits of detection, as well as fast response times. I don't want to spend too much time on how we make these and how we characterize them, but I'm happy to answer any questions at the end of the talk about this electrode platform. So as I mentioned earlier, the electroactive material, it's a pyrolyzed photoresist film. As such, is very similar to glassy carbon. It's hard, it's inert, it's conductive, and most importantly for us, it's compatible. In addition to that, this electrode platform actually is transparent. So what that allows us to do is actually introduce this electrode platform onto a microscope where we can do simultaneous imaging of the cells and look at their optical densities, whether they're alive or dead, while we're performing our electrochemical analysis and detection of Piocyanin, which is pretty awesome. So now that we know this platform, before I show you any of the data, I'd like to introduce the technique that you're going to see basically throughout the rest of this talk, and that is square wave voltammetry. This is one of the pulse voltammetric techniques that we use in electrochemistry, and it's considered to be one of the most sensitive voltammetric techniques. So here I'm showing very simple image of what happens as we apply potential over time. So we have a starting potential and an end potential, and we actually step it with this kind of a staircase waveform, if I can call it, till we get to this end potential to get something that actually looks like this. So we basically are sampling the currents at 2 times during this technique. We sample the current right here, right at the end of the forward pulse, and then right at the end of the reverse pulse. So if we start looking at the responses that we get of the current as a function of the potential, we can look at the forward versus the reverse current response. However, we never view these in isolation, but rather we just plot the difference in current, which is the forward minus the reverse. So because of the places at which we sample this current, if you look at the current profiles over time, we actually, at this time points, get the highest decay of this charging current that contributes a lot to the noise. So there's two types of currents that we care about in electrochemistry. One, it's Faradaic, and then we have the non-Faradaic current. So Faradaic current is resulting from processes that involve electron transfer on the electrode surface and processes that we care about, and then everything else that's contributing, it's called the non-Faradaic charging or capacitive current, which actually contributes a lot to the noise. So because of the sampling times in this technique, we actually get very high sensitivity. It's very fast. It takes seconds, not minutes or hours, to acquire a square wave voltammogram. And this peak height, we can actually use it to build calibration curves from which we can determine unknown concentrations from unknown samples, which we love to do in analytical chemistry. So just to give you an overview, if you're familiar with anything electrochemistry or chemistry in general, you probably have heard of CV, which stands for cyclic voltammetry. So in this example, I'm pretty sure this is a cyclic voltammogram of hydrogen peroxide, where you can kind of see the redox peak of the anodic and the cathodic contributions, but it's really hard to tell where these peaks occur. So if we take the same sample and run a square wave voltammogram, we get a nice sharp peak, and this has to do again back with the fact that square wave is a highly sensitive technique. So I don't want to spend more time on this, but this is the technique we're using, and it's very commonly used in biological systems to really quantify in electrochemistry. So now that I explained all of this, we started using our TCUAs to specifically look and detect Piocyanin. So using a range of biologically relevant concentrations of Piocyanin, which have been previously determined from biology studies, we did increasing concentrations ranging from 1 to about 250 micromolar, and you can see that this square wave current responses increase as we're increasing the concentrations of Piocyanin. Taking this peak currents, then we can construct a calibration curve by plotting the current as a function of the concentration. And from this, then we can determine concentrations from our bacterial cultures where we don't know the concentrations actually. So based on this data, we determine our limit of detection to be 1 micromolar and our linear dynamic range to go from 1 to 250 micromolar. So just to give you a little bit of comparison with other studies in the literature that have reported sensors for Piocyanin detection compared to our TCA's method, you can see that our limit of detection, there's some that are lower than ours. However, in this present case, this limit of detection actually falls within the ones that are determined from in vitro environments directly from patients. So with that, we actually are pretty happy. And in this particular case with Piocyanin, it's not necessarily about achieving the lowest limit of detection, because the lowest that's been isolated from patients and measured is 1. What matters more, it's actually with this linear dynamic range, because the concentrations they've isolated from patients have been found to range from 1 to 180 micromolar. And at present, we're actually the only electrochemical sensing platform that can detect beyond 100 micromolar. So that is pretty exciting. In addition to all of these advantages, our TCA platform, it's relatively cheaper than some of these other electrodes, such as the boron doped diamond. And finally, as I mentioned earlier, we actually have the advantage of having this transparency of the electrodes, allowing us to do optical density studies and allowing us to basically watch the cells as we're performing electrochemical analysis. So with that, we did a proof of concept study to prove this advantage, where we took two different strains of Pseudomonas aeruginosa. So we have PA14 and PA11. PA14 is a wild type laboratory strain, which is considered to be highly virulent, and it produces high levels of Piocyanin. So this is what biologists commonly use in studies, and something you will see in a lot of microbiology papers that deal with Pseudomonas aeruginosa. And the other strain we used is PA11. This is a clinically relevant strain from isolated from the lungs of cystic fibrosis patients in the UK, and was brought to the US by my collaborator, Sophie. So basically what we can see, here's square wave voltammograms that we acquired while simultaneously imaging the cells. These are bright field images, and you can see the cells swimming. Some of them are blurry, especially this one here. So with these images, you can quite tell the difference between the PA11 and the PA14 strain. We can look at their cell densities from these images, but more importantly is that with EChem, we can actually quantify the production of this Piocyanin virulence factor. And in both cases, you can see these are just different growth conditions for the cells, slightly different growth conditions, but you can see in both cases that this laboratory strain is not as virulent, and it's not producing as high concentrations as the one isolated from patients. So these are the concentrations we determined for the PA11 strain, and these are the one for the wild type laboratory strain. So now that we got this promising initial results, something I became really interested in for the last 2 years almost is, can we use this platform to do real-time electrochemical monitoring of Pseudomonas aeruginosa metabolism as a function of more complex growth environments? So everything I showed you so far has been done in very simple buffered solutions, phosphate buffer, which doesn't necessarily contain all the biological species that can have an impact on the electrode surface and often cause biofouling. So all the sugars and proteins, they're not present in the simple buffers. So what we really wanted to do is take different simulated growth media and see if we can electrochemically monitor the Pseudomonas aeruginosa cultures. So for that, we chose two broths. These are lysogeny broth and tryptic soy broth, which are going to be labeled as LB and TSB for the rest of this talk. These are two very commonly used bacterial broths in microbiology labs. The truth is there's endless possibilities when it comes to growth media. However, these are the most commonly used ones, so we decided to stick with them. So just to remind you is I became really interested in seeing how does the dynamics of this Pseudomonas aeruginosa biosynthetic pathway change as a function of the growth environment. So what we did is we started doing this real-time electrochemical studies. So here I'm showing you a 3D plot where you see, these are all square wave voltammograms, by the way. So you can see the current as a function of the potential as a function of the time. So these studies were performed over the course of 48 hours. The reason why we selected this time window is that in hospitals, typically it takes about 2 to 3 days to just identify the species that the patient might have. So we wanted to understand what's happening in this 2-day window as the cells grow. So what you can start seeing is we have Piocyanin production during the first initial hours, and then it's increasing over time, and at about 21 hours of bacterial growth, it reaches its maximum concentration, and then it's slightly decays and plateaus. The other interesting thing we started observing in this data is that we started getting this additional peaks, which initially we were not aware of what they are. And I'm going to tell you more about how we identified what these peaks are during more intermediate stages of bacterial growth. So that's this peak right here, as well as the shoulder peak during later stages of growth and more negative potentials. So if we take a look at the same study performed in the other media, TSB, we actually start seeing significant differences in the dynamics of this pathway. Namely, there's higher concentrations of phenazine species produced in this media. And in addition to that, during intermediate stages of growth, in this particular case, the current responses for this 5-MCA peak are actually higher than Piocyanin at certain stages of growth, which is not the case here. And then we have all these other peaks that are showing at more positive potentials, which are not present right here. So what we really wanted to go ahead and understand is what in the media might be contributing to these differences, because if you talk to our microbiology collaborators, they will tell you the cells should grow similarly in both media. And yes, they do grow similarly in terms of their optical densities. The phenazine production of this virulence factors, it's actually rather different. And there's something in the environment that really impacts it. So to characterize this growth media a little more, we actually decided to do mass spectrometry studies, in particular this DESI, which stands for Desorption Electrospray Ionization mass spectrometry of both media. So in the top here, these top two graphs are showing you the positive ion mode mass spectrometer. So this spectra actually just generate positively charged species, or you can perform the experiment also in the negative ion mode where you create negatively charged species. Based on whether you protonate or deprotonate the species, we can actually identify what kind of species we have in the growth media based on their fragmentation pattern and masses. So there's a lot happening right here, and this is a really busy slide to look at. So I want to point to two big differences that we noticed in our data, in particular in the positive ion mode. If you take a look at this choline species, choline is a water-soluble vitamin which bacteria consume to grow and produce virulence factors. So you can see in TSB medium, the relative abundance is higher compared to LB media. And another difference that we observed that was significant given all these results in the negative ion mode was the difference in relative abundances of this hexose species. Hexose just refers to a 6-carbon sugars such as glucose and other derivatives. And you can see that the relative abundance of it in TSB media, it's much higher compared to LB media. So this is actually more food for the cells to consume, which might be explaining why we have higher production of phenazine compounds in TSB media compared to LB. This is obviously not the full picture, but it provides some explanation of what might be happening in this complex environment. I also want to note that this mass spectra were taken only in a range from 100 to 400 MZ. And if we extend this window to about 1,000, we can even look at all the other species like lipids and proteins that are changing, and it's rather very complex. So now that we understand a little better of what might be contributing to this difference in phenazine production based on the media that the cells are grown in, we wanted to go ahead and really identify what these peaks are. So initially, based on its electrochemical signature around negative 0.256 volts versus a saturated Kelmo electrode, Piocyanin shows up right here. So we knew that's the peak of Piocyanin. However, we're not sure of what the rest of the peaks are. But to do that, we again turned to mass spectrometry because it's a really good technique to identify qualitatively what species are based on their masses and their fragmentation patterns. So first, we again did this desorption electrospray ionization mass spectrometry, where in this case, we actually coupled it with a mass spec imaging techniques. So alongside of our electrochemistry experiments, we also were performing this mass spectrometry imaging techniques. This is just a small droplet of the cellular culture that we're detecting from. And this is just the scan that the image was created from. And you can see the media obviously does not have any piocyanin. However, when we have cells, we start seeing production of piocyanin, both LB and TSB media. And this different coloring scheme is just false coloring to indicate the intensity, which actually refers back to the relative abundance we're seeing. So it's just a scale, and you can see that over time it increases. So it's a very good qualitative way that confirmed our quantitative electrochemical data. So we were actually able to detect piocyanin at almost all hours of growth. And with that, we actually assigned this peak, which we can see during each hour where we performed this experiment, to piocyanin. The only other species that we're able to detect using this desorption electrospray ionization method was 1-hydroxyphenosine, which I've labeled as OHPHZ. This is actually a side product which was not shown on the pathway, but it forms as a decomposition of product of piocyanin under slightly basic conditions. And we were only able to detect this species during later stages of growth. So that, again, we assigned it to this peak based on what we're observing into the electrochemistry data. In addition to that, we still have this peaks right here, and we had a lot of hard time detecting and identifying what they are. There's been a bunch of proposed pathways by microbiologists on what kind of phenosine species are involved in this biosynthetic pathway, but we're not sure what the exact identity was. So the best, after trying various other mass spectrometry techniques, we were finally able to detect it. And before I tell you about how we're able to detect it, I first want to tell you why it was so difficult to identify this peak. So if you look at the last step of this biosynthetic pathway that I showed you at the very beginning of this talk, this conversion of 5-MCA to piocyanin, you can see that this reaction is highly dependent on molecular oxygen present. So here, I performed a very simple square wave voltammetry experiment where at 5 hours of bacterial growth, I took a square wave voltammogram. So you can see the peak for 5-MCA and then piocyanin. If you leave this culture exposed to air for 1 hour, what happens is that the current response for 5-MCA almost completely diminishes. And then the current responses for piocyanin increase, which actually further confirm that this is the step and it's dependent on molecular oxygen. So with desorption electrospray ionization, you actually need to dry your sample out for an hour before you apply this charged spray of solvent to analyze. Otherwise, you just basically blow off this droplet of bacterial culture and you're not able to analyze anything. So after I saw this, with electrochemistry, I started understanding why we actually can detect this species with our DESI approach. So I went and talked to more of our mass spec collaborators at UT Austin, and Chris Crittenden, actually from the Broadbelt group, helped me finally get the mass spectra in both the positive and negative ion mode of this 5-MCA species. And for that, we used a technique called electrospray ionization, but we used a nano version of it where you use a very small nanometer size needle to directly inject your sample into the mass spectrometer without the need to wait and dry for 1 hour. So finally, we're able, based on this fragmentation patterns, to confirm that this truly is this 5-MCA species. So giving us even a better picture of what this phenazine biosynthetic pathway in Pseudomonas aeruginosa looks like. So we assigned this peak right here because we performed the studies during the hours where we're observing it into our electrochemical data. The only other peak that remains is the one right here at very positive potential relative to Piocyanin, obviously. And at present, the identity of this peak remains elusive. We believe that it might be a species that is a derivative of 5-MCA, or it might be another species that is highly reactive to oxygen. And in the future, studies need to focus on identifying what this peak actually is. So it's very interesting. So now that I got a better picture of what's happening when we're growing this Pseudomonas aeruginosa monoclonal cultures, the next question I became interested in looking into is, can we perform these studies in even more complex environments where we have polymicrobial environment or more than just Pseudomonas aeruginosa? So with that, we decided to introduce two other common pathogens that are commonly found around Pseudomonas aeruginosa, and those are E. coli and Staph aureus. And the question we really wanted to look into is, how do we impact the dynamics of this Pseudomonas aeruginosa phenazine production in this even more complex biological environment where we have other species where they might be competing for food or having more synergistic relationships, and does this depend on the growth media? So I will show you a very short summary of the data from these studies because it's actually a lot of data to look at. But if we look at the concentration of Piocyanin quantified in this polymicrobial cultures, in comparison to just the Pseudomonas aeruginosa cultures, we actually start seeing a lot of differences. In particular, when Pseudomonas aeruginosa is grown on its own, we observe highest production of Piocyanin in LB and TSB media. However, when we introduce Staph aureus, it doesn't seem to have a huge impact, though there is differences in the concentrations produced, and in LB during later stages of growth produces even higher amounts of Piocyanin, which is not the case in TSB media, where the concentrations of Piocyanin are lower. If we then take a look at the cultures that contain E. coli, so this is Pseudomonas aeruginosa with E. coli, and this is the all three species together, we actually see that the presence of E. coli seems to diminish completely this production of Piocyanin, and that's even more obvious in this TSB media, which is more nutritionally rich, as we saw from the mass spectrometry data. So based on some evidence in the literature, E. coli produces a molecule called indole, which some microbiology studies have suggested to directly interfere with the phenazine biosynthetic pathway in Pseudomonas aeruginosa. We actually tested for the presence of indole using a qualitative biochemical test and a reagent, which actually turns pink if you have indole in your E. coli cultures. And that seemed to be the case. So we believe that that might be contributing to this diminishment of production of Piocyanin when E. coli is present in these cultures. So with that said, this actually remains a very open question and something that needs to be explored even further in the literature. And another thing I'd like to remind you of is that this species in some of the media might be also competing for food and nutrients. So E. coli is actually particularly known to grow rather fast, at really fast rates, and consume nutrients rather fast than other species. So that might be another reason and explanation why we're seeing this reduction, Piocyanin production, and here it's almost zero. So while we don't have a full explanation of what's happening, the very exciting thing is that our platform actually works for detection of Pseudomonas reginosa in very complex biological environments that closely resemble those of real environments. And at present, we have an electrochemical platform that can not only measure and quantify the differences in final products produced, but also can detect differences in concentrations of intermediate species. Obviously, I'm not showing all the data here, but we've quantified the MCA concentrations as well as the 1-hydroxyphenazine concentrations, and both of them are impacted by the presence of other pathogens. So with that, I want to leave you all with one big question that really comes out of all these studies is, based on all these environments that we use in laboratory studies, we actually need in the future to focus on analytical standardization of this simulated growth media and develop growth environments that closely represent chronic wounds or the cystic fibrosis lung, that will provide an even more realistic picture of what's happening with as infections develop in the human body. With that, specifically determine which specific components and in what concentrations in these biological environments contribute to changes in the dynamic metabolism of pathogenic bacteria. So this will take a lot of work, but obviously it's necessary if we want to study these development of infections in a more meaningful way that closely represents what's happening in the body. With that, I would like to thank all the people who have helped me throughout my PhD journey. First and foremost, my PhD advisor, Professor Keith Stevenson. He's been absolutely amazing mentor, and I'm extremely lucky to have had the opportunity to work with him. He actually was in Skoltech for most of my PhD career, and he's shown me that we can make long distance mentoring work if we're committed to each other and we respect each other. So I'm very thankful for him. Also, members of the Stevenson group, in particular Janine Elliott and Jonathan Dewey, who trained me a lot on the electrochemical techniques, and Jonathan, who actually did a lot of the initial work on developing this TCA platform, which has been the basis for my PhD. My mass spec collaborators, Marta, Professor Olivia Eberlin, and Chris, for being so engaging and a pleasure to work with, all of my funding, and the rest of you who are listening tonight. I'm happy to take any questions. Thank you.
All right. Thank you very much. As expected, it was a fantastic talk. While I wait on chat to get any questions in that they might have, sometimes it takes a minute to get them typed up. I'll go ahead and ask a couple, if that's all right. So the first thing I'd like to ask is, with the electrode itself, you said, and I may have missed this. You said it has a quartz plate, right? So it's on a quartz slide. You put a photoresist down, and then you have a.
I'm actually gonna pull a slide.
Yeah, please do.
I can show you what it looks like exactly. So here's
Okay.
to fabricate them. So basically we have, yes, we start with a quartz slide, and then we apply photoresist, and then we pyrolyze it. Then we basically drop cast a solution of polystyrene spheres and methanol. So they organize themselves into this hexagonal organization as the solvent evaporates.
Okay.
They stick to each other. After that, we do atomic layer deposition to apply 10 nanometers of aluminum oxide, and we do this at 80 degrees, because if we go higher, we actually melt the polystyrene spheres.
Okay.
Upon that, we then do various sonication baths to remove and melt this polystyrene sphere. So what you're left with is basically this areas of pyrolyzed photoresist film, which is our electrode. And then the rest of it is the aluminum oxide layer. And here you can see this top view where D is this diameter of the polystyrene sphere.
Sure.
And then A is our radius of each individual electrode. There's a mathematical model where we can predict the sizes. And we also took AFM images to confirm what the sizes are. So this is just top view where you can see the little individual electrodes in this array. They're all connected in a parallel circuit. So if we take a look at the cross-sectional area of this AFM image, right here, we basically can determine the radius, which is about 0.097 micrometers. So we're actually down to nanometers, which is pretty exciting. So a lot of people ask me, why the hell do you need spheres? Why can't you just have a plain carbon electrode? And I have this actually on the next slide. So if you basically, we've tried this fabrication with various different sizes of polystyrene spheres. So these numbers are actually just representing the diameters of that. And T macro is just plain pyrolyzed photoresist, like carbon electrode.
Okay.
So if we look at the radius and the carbon areas, obviously here we have 100% carbon area, and here with the 1.5, we have highest percentage of carbon area. But by introduction of this alumina oxide layer, it actually acts as a dielectric. So any charging you get from double layer capacitance from just water sitting on the carbon electrode surface is actually greatly diminished by the presence of this aluminum oxide layer, which organizes the charge. So if we take a look at the signal and the noise separately at each of these electrodes, in particular, this is the no array electrode, you can see the noise is significantly higher.
Right.
If you look at the difference in dielectric constants between water and alumina, that explains why we see this difference in noise. And our signal basically is just goes back to the exposed carbon area, and it's highest when we use this smallest kind of diameter. So we get highest carbon area, and we're able to achieve the best signal to noise ratio with this electrode geometry. So.
Okay, that's interesting. And I'm sorry. And the quartz slide underneath is. Oh, I'm sorry. No, you have the pyrolyzed film that provides the conducting layer. Is that right? Am I understanding that right?
Mm-hmm.
Okay.
Absolutely.
All right, I got you now. All right.
Yep.
So do you foresee this being used in like in terms of applications as an analytical device? Do you see this being somewhere maybe a doctor is trying to determine if somebody has, say, cystic fibrosis, or is there a different, like, how do you see this being used?
That's been my long kind of vision,
Okay.
and hopefully I will get to work on this at some point later in my career, or someone else in Stevenson's group, hopefully. But the idea is to basically, yes, apply this in a hospital, and really, because electrochemical sensors, in addition to providing quantitative ways, they actually are relatively cheap compared to everything else, and they're relatively easy to use.
Mm-hmm.
So you might think of using this in the hospital, where you can have your patient suffering from cystic fibrosis. You can isolate a sample from the lungs, and instead of doing all this cell isolation and plating on basically this agar plates that have specific growth media for Pseudomonas, you would be able to basically just identify it based on whether you have Piocyanin in production or not. Another area where it can be really interesting is so this phenazine virulence factor production. There's recent studies that have shown that it actually helps bacteria develop antibiotic resistance mechanisms. And at present, we don't know what concentrations of Piocyanin, like what is the limit where they become antibiotic resistant.
Okay.
So that's another area where you could use it in the future, hopefully when we have more information on what specific concentrations are, because if you're past that limit, then you just end up giving antibiotics to the patient without helping the patient.
Yeah.
You're actually helping the bacteria develop more antibiotic resistance mechanisms,
Okay.
so it's not good.
Yeah, that's interesting. Do you think this could be used? Like, is this kind of? I'm trying to think of the right word. I guess modular is not quite right, but could you basically change out for different applications? Like, if you wanted to look for a different molecule or a different product, I guess basically, is that something that could be done?
Absolutely. Absolutely. I mean, this platform is very easy to modify.
Hmm.
So we've actually done detection of hydrogen peroxide and nitric oxide
Okay.
on these molecules. So nitric oxide actually is particularly interesting because it's produced by your immune cells in
Hmm.
defense to this pathogenic infections.
Okay.
And for that, we actually needed to further modify the electrode with gold nanoparticles to increase this electrocatalytic activity.
Hmm.
And we needed to apply on top of it a cellulose acetate membrane with a pore size of about 6 angstroms that basically filters out all this other biological species
Okay.
and really selective for just nitric oxide. So phenazines, which I mostly talked about during this talk,
Hmm.
they're actually kind of beautiful in a sense that the potential window in which they show up, it's in the negative potential window.
Hmm.
There's not a lot of the common biological species like ascorbic acid, glucose, uric acid,
Yeah.
hydrogen peroxide. They're actually in a positive potential window. So when we started to look at nitric oxide, this became a huge problem because we were getting all this other interferences, and introducing that cellulose acetate membrane greatly helped. So like we could really get down to just nitric oxide, and the only other species that we're detecting is hydrogen peroxide, but with square wave based on the potentiodynamic signatures, you can distinguish one from the other because they're about 100 millivolts apart.
Okay.
Mhm.
That's really interesting. Yeah, that's it's nice that that can be kind of adapted on an as needed basis.
Yeah, and something, so Nets, who talked last week,
Hmm.
platform, he talked about aptamer modification for
Yeah.
specific molecules, and that's something that we have tried to do on these electrodes. Unfortunately, I graduated, so
Okay. Yeah.
with the next person to do the work, but it's a very easy to fabricate and very easy to modify electrode platform, I would say. It's very
That's really nice.
Mhm.
That's fantastic. Um, so what would you say are the limitations on this? Like, where, I guess, not necessarily where is it bad, but where do you see the places for it to improve as a device?
I think one place, and this actually like goes back to what I presented on today, is finding a way to couple it with portable mass spectrometer would be amazing,
Hmm.
because oftentimes we can detect, what are electrochemically active species, but then we don't know what is happening in the environment. And
Okay.
looking at those molecular signatures of this growth environment greatly helps. So that's something we've talked about with my mass spec collaborators. Is there's some studies in the literature where they've combined electrochemistry with mass spectrometry, but that's where I would see it. And obviously, the one of the limitations is when you have species that are not electrochemically active,
Hmm.
then you probably need to use like an electrochemically active molecule as a tag.
Yeah,
And
that makes
look at
sense.
specifics of the chemistry, but that's where I would see are the limitations. Actually, the biofouling on this platform has been amazing,
Hmm.
which I know a lot of the other electrochemistry sensors for this. it's actually a big issue that they see.
Okay.
However, our platform, we have over 10,000 electrodes in this area that we work with.
Okay.
So, and most of it is dominated by this alumina oxide layer. So we actually, if you look at the current responses and just the buffer that were around the experiments
Mm-hmm.
before the 48-hour experiments and after, they're almost identical. Like we
That's
can't
really
see
good.
differences. So that's been a very good indication of the biocompatibility.
Yeah,
So
that's a really important thing because it's, you can have the best electrode in the world, but if it, if you can only use it once or twice, that's not, it's still okay, but it's not great.
yeah, and that's something like, a lot of the platforms that are being developed are this disposable carbon
Mm-hmm.
paper, and it's nice, but there's a lot of variation from one to the other.
Yeah, absolutely.
So the reason why I like this platform is, you can truly monitor like this growth over really long periods of time and not get significant biofouling. So
Yeah. Oh, that's fantastic. There was something else. So, what is the sort of the minimum time resolution on the technique, at least right now?
you mean how long it takes?
No, so how like let's say there's a reaction or a product that's forming that forms very quickly.
Mm-hmm.
Is that something that you could capture? Like I know you showed from 0 to 48 hours, and you were kind of doing an hourly measurement, but
Yeah.
let's say it formed on a scale of like a minute or two. If you knew that, I mean, obviously that's something you would kind of have to have an idea of beforehand. But if you knew that, is that something that you could actually track in real time as well?
Yes.
Okay.
So we actually, I don't have the data on the slides, but we've determined the response times on these electrodes,
Mm-hmm.
and they're about 150 microseconds.
Oh wow. Okay.
Rather fast. Like we
Yeah, that's
can,
very fast.
yeah, absolutely. And with a combination with square wave, you have the instrument and the sensor itself.
Right.
One
Right.
of
Right.
the best of both worlds. But yeah, it's rather fast compared to the T macro electrode, where
Mm-hmm.
it's just the carbon, the non-array. I'm recalling, I don't know the exact numbers, but it was on the order of 1500
Okay.
microseconds. So
Okay.
quite large difference.
And that was due to the double layer that forms the
Yeah. Like if you
Faraday.
start looking at the capacitive and
Yeah.
resistive components
Mm-hmm.
and what each is contributing, and if you do like a kind of an IT curve, you can basically determine this response times with like a response factor correction.
Right. Okay.
Mm-hmm.
That's fantastic. This is really interesting stuff. I said,
Mm-hmm.
this is
Thank
it's
you.
between you and Nets. It's been very interesting to see the combination of fundamental and applied work that's going on when it comes to biological species. Because
Yeah.
my background is all chemical, and so
Yeah.
it's this is all it's very incredible to see, really.
Yeah. And I mean, these environments are very like complex.
Yeah.
Most of the time, we don't even know what is happening. And the fact that we can quantify and measure and
Mm-hmm.
get better understanding, it's truly fascinating to me.
Yeah, it really is. Like, and I especially like the way that you could see the changing over time.
Yeah.
Because that's one, you can take a snapshot and that's fine, but you start taking multiple pictures and all of a sudden you see the difference as the reaction actually proceeds, and it's really cool that you can see multiple products form and then decay away as well.
Exactly. Exactly. And I think we have a really high sensitivity on this electrode platform because I've seen papers where they developed like platform for Piocyanin, does just like a carbon fiber electrode, for example.
Mm-hmm.
And they would grow the cells for 48 hours and then do the test or like at 12 and then 40 hours. And they're not really able to see those other peaks.
Yeah.
They see Piocyanin, but they don't see the other peaks. And that's something that actually got me really excited for this work.
Oh yeah, absolutely.
Because going in, I had no idea we're going to get this additional peaks in the data. I just did the experiment to try and see what I get. And
sure.
Then obviously, you need mass spectrometry, because like that's one of the limitations. But electrochemistry, we can distinguish and say we have different species on the electrode surface, but oftentimes we might not know. And that's where we need things like mass spectrometry and like IR or Raman at times to identify what species we might have. So
yeah, I got you.
It's combined techniques.
Yeah. Oh yeah. It seems like you always have to get. It's very rare that you can use just one.
Yeah.
That's fantastic. All right, so I'll give last chance for any questions from the chat. And it looks like I think that's going to be it. So, Olja, I would like to thank you for coming. Again, this is Dr. Olja Simoska from the University of Utah. Like I told you, a great researcher, and presented some wonderful research tonight. Olja, thank you for coming on. I very much appreciate it. And